Hi everyone, welcome back. Today we're going to be discussing organ transplant, which is covered in the book in chapter 15, sections 29 to 38. And I'll again be referring to figures in the book, and you'll see the figure numbers enlarged here so you're able to refer back to your textbook. So today we're going to be talking about first an overview of organ transplantation. And we'll also be talking about the important immune responses against grafts that lead to graft rejection. In particular, we'll be discussing T-cell and MHC-mediated responses, as well as the meaning of an alloantigen and alloantigenic peptides, and also the development of anti-graft antibodies. We'll also talk about different types of rejection, such as late-stage graft failure, and we'll discuss as well the roles of regulatory T cells. And finally, we'll wrap up with a couple examples, including graft versus host disease, and an example using the fetus as a tolerant allograft, so as an allograft that's non-self but is typically not rejected. So we have discussed throughout a few different lectures this idea of tolerance. And this is a nice chart summarizing some of the different mechanisms of tolerance. So tolerance means that the immune system has to learn to recognize self-antigens as not harmful and not activate an immune response against them. And when this fails, you see responses such as autoimmunity. And this is also really important in the discussion of organ transplantation because the organ that's transplanted is non-self and so therefore is not tolerized. However, we'll discuss some ways that the chances of rejection can be reduced and how these immune mechanisms can be modulated. So we'll start out talking about an example of an experimental skin graft in mice. So first we see this skin graft between two syngeneic mice. So these are mice with the same genetic background. So you can see here that's going to be represented by the different colors. So if we have skin from a yellow mouse transplanted onto another yellow mouse, this graft is tolerated and not rejected. However, if you have skin from two different mice, in this case a yellow mouse transplanted onto a blue mouse, this graft is going to be rejected. And this is because that graft is recognized as non-self. And we can see in these experimental processes that this seems to involve the adaptive immune system. And we can tell that because the first time the graft is rejected, this process takes a while, takes 10 to 12 days. However, if another graft is given from the same donor, this graft is then rejected much faster. So this is typical of an adaptive response. So we can then narrow down which parts of the adaptive response are important by transferring cells from a mouse that is already sensitized to the donor skin graft. So if we take T cells from that blue mouse with the yellow skin graft and transfer it to a naive blue mouse that's never seen the yellow skin, then this can actually lead to the rapid rejection that we see in the mouse that's already been tolerized to this skin graft. So this transfer of T cells helps us to know that it seems like T cells are mediating this graft rejection. And so, as I said, syngeneic grafts survive. So if the graft is from an individual that has the same genes, then it is going to be recognized as self. However, in differently genetic background animals, we have something called alloantigens. And these are antigens that vary between members of the same species. So for example, the yellow mouse and the blue mouse. In allogeneic grafts, the first rejection, like I said, is in 10 to 13 days, and the second is about 6 to 8 days, indicating an immune response, in particular an adaptive response. And we also see rejection primarily due to a response to non-self MHC, which we'll discuss. And actually, MHC, the name Major Histocompatibility Complex, comes from experiments of transplantation. This idea of histocompatibility, or the ability of a graft to not be rejected, is due to the similarities or differences in MHC molecules between individuals. And in particular on tissue grafts, this is due to MHC class 1. And rejection still occurs slowly if this MHC is matched. So if you have the same MHC between the donor and the recipient, you won't have this acute dramatic graft rejection. However, as we'll talk about, the graft is still very slowly rejected due to other differences between these individuals. So like I said, when MHC is matched, rejection can still occur. So this is due to alloantigen peptides bound to the graft MHC. And in this case, because individuals will all have slight variations in their genetic background, these differences, when they are presented on MHC, are recognized as non-self by the recipient immune system. These polymorphic proteins lead to these non-self unfamiliar peptides, and these are known as minor histocompatibility agents. And in response to minor histocompatibility antigens, the body mounts an immune response that's very similar to that seen with a viral infection. And one really good example of this type of rejection is with genes on the Y chromosome. So because males are XY and females are XX, if a graft from a male, or XY, is transplanted into a female, that female has never seen the antigens on the Y chromosome, and therefore they'll be recognized as non-self. And this is a female anti-male rejection response. However, it's due to these other alloantigenic peptides, not the MHC molecules. So again, if we refer back to our skin graft mice, so here again we have our syngeneic transplant as well as our allogeneic one. However, now we're adding in a mouse that only differs in the minor histocompatibility antigens. And here, this graft is rejected much more slowly. So because this rejection is due just to the peptides on those MHC molecules, it doesn't necessarily lead to a rapid immune rejection response. So there are two different types of recognition of alloantigens. The first that we'll talk about is when the organ grafts contain donor antigen presenting cells, and this is known as direct allorecognition. So in direct allorecognition, donor APCs will migrate to the lymph nodes through the blood in the recipient, and this is opposed to the typical migration through the lymphatics, and that is because in a transplant, the lymphatics are disrupted, so the only way for those APCs to get to the lymph nodes is through the blood. Then we'll see that these APCs can activate host T cells with the corresponding T cell receptors, and these alloreactive T cells will then attack the graft. So we can see in this figure an example of an organ transplant of a kidney. So those dendritic cells, seen in yellow, in the kidney can leave through the blood, and they go to the lymph nodes here and present that antigen to T cells, which then can migrate back to the kidney and lead to destruction of the tissue. And this rejection is much slower if these donor APCs are first removed. However, antigen rejection can still occur even if the donor APCs have been removed. And this is known as indirect allorecognition. And this involves the APCs from the recipient. And here, those APCs from the recipient can go into the organ and take up those allogeneic proteins. And they present those peptides on self-MHC to T cells and activate those T cells. And this can also activate cells such as macrophages, which leads to more gradual tissue injury and fibrosis. However, T cells that are activated this way through MHC2 presentation can only really provide help for those antibodies against the alloantigens. So they provide help to B cells and lead to an antibody response. However, direct T cell attack can only occur when T cells recognize the graft MHC directly. So here we have a figure showing the difference between these two responses. So on the left, we have our direct allorecognition. And here, the CD8 T cells from the recipient can directly recognize peptides presented on the donor MHC. However, in indirect allorecognition, we have antigens that are slowly taken up, processed, presented on MHC to T cells. And this is a much slower, more gradual process. On the other end of the spectrum, there's graft rejection response known as hyperacute graft rejection. And this is due to the presence of pre-existing alloantibodies. And typically these antibodies are against the blood group antigens, so these are the antigens that differentiate the A, AB, B, and O blood types. Or they might be antibodies against the non-self MHC. And this is a response that triggers complement within minutes of the transplantation, so still during the surgery. The antibodies against these donor antigens can bind the endothelial cells within the graft, and this can eventually lead to a buildup of complement seen here, which can block those blood vessels. And if enough blood vessels are blocked, the graft cannot receive enough oxygen and it will die. And this is usually avoided by matching the ABO blood types and cross-matching, which is a process where the antibodies of the recipient are checked against the donor white blood cells to make sure there's not going to be any reaction there. And interestingly, it's not very well understood, but there are some organs, especially the liver, that are resistant to this hyperacute graft rejection. It's really not known why that is. You might hear discussion of xenografts in talks of organ transplantation. So a xenograft is a transplant into a human with an organ of animal origin. And this is discussed often because there's a dramatic shortage of donor organs. And so the ability to use organs from an animal, especially a pig, would help reduce this shortage. However, we run into problems with this hyperacute graft rejection. And that's because most humans have antibodies against alpha-gal, which is a carbohydrate on the cell surface of other mammals. And these antibodies can then recognize these antigens on the cell surface, leading to hyperacute graft rejection. So we've discussed immediate graft rejection or acute rejection. Now, one of the big problems with organ transplant rejection is this late failure of transplanted organs that occurs over time. And this is really the main issue that affects organ transplantation these days. And that's because now when someone receives a donor organ, they're also given immunosuppressive drugs, and those organs are matched as well as possible with the major and minor histocompatibility complex antigens. And this helps reduce that acute rejection. However, this long-term graft survival is still problematic, and this is due to chronic rejection. And what this means is that you might see things such as chronic allograft vasculopathy, which is a concentric arteriosclerosis of graft blood vessels. And I'll show you what that means in a second in a figure. But this is most common in highly vascularized organs such as the heart and kidneys. And it leads to reduced blood flow to the organ and can also lead to fibrosis. This is thought to be caused by very minor but recurrent acute injuries to the organ. So, for example, maybe some allospecific antibodies or T-cell response, or it could also be due to an inflammatory response that's induced at the time of surgical transplantation. And immunosuppressive therapy can actually also, besides suppressing the immune system, lead to this late failure of transplanted organs. And that's because some of these therapies can promote protein deposits in the smaller vessel walls.